So good morning, everyone. Thank you so much for joining our application seminar focused around screening. My name is Cameron Skinner, and I'm a product manager at Millennium Science. Before launching into our talk today, I thought I would quickly run through some points about Millennium Science and then introduce our speakers. Millennium Science was established in 1999, and for 20 plus years, it has expanded to be a leading source for most advanced biomedical research technologies in both Australia and New Zealand. We recently celebrated our 2-year anniversary merging with JOMA Life Research, which gives us access to a vast portfolio of reagents, which also includes screening reagents. Millennium has also been a part of the screening community since 2006, and we are proud of our association with today's speakers and many more researchers. On the screen now, you'll see a couple of different suppliers we supply for. With the many brands that we support, this means that Millennium contributes and supports the science, in many different ways and areas. As today's focus is around screening, I thought I would demonstrate some of the standout areas within this application that we support. Everything from automation with instruments, reagents like compound and gene modulation libraries, and services like engineering cell lines and also cell panel screening. So to kick this off for our application seminar on screening, I'm proud to announce our speakers today. Our first speaker will be Dr. Amee George, Manager of the ANU Centre for Therapeutic Discovery, and also a Fellow for ACRF, and Associate Professor Kaylene Simpson, the Head of the Victorian Centre for Functional Genomics at the Peter MacCallum Cancer Centre. So I thought I would kick it over to Amee to start our chat.
Great, thanks Cam. I'll just share my screen. Thanks everyone for your attention today. Thanks for the introduction, Cam. Just wanted to start by saying I don't have any disclosures to report, but really for this audience, just wanted to introduce what high-throughput screening is. It really enables us to, on a large scale, be able to efficiently measure and test the effects of different agents or conditions in our biological chemical assay. And it's a really important tool that we use in drug and biomarker discovery. And what it really helps us to do is to, move from testing individual targets, which you might be doing currently in your own laboratory environment, through to multiple target testing, ranging from screening in 96-well plates right through to 1536-well format. And so for us to be able to do this, there are a number of important requirements. We do require robotics and automation platforms, firstly to be able to cope with the sheer amount of plates that we screen, but secondly also for robustness of the assays and maintaining reproducibility. Of course, there's screening libraries, as Cam mentioned, biological and chemical libraries, assay readouts, and of course we need streamlined data analysis pipelines and bioinformatics to be able to cope with the sheer amount of data that we generate with these types of experiments. And so I just wanted to briefly show you my facility, which is the ACTD. This is a picture of it here. We're a fully automated high-throughput screening lab. We opened back in 2017. We're a member facility of Therapeutic Innovation Australia and also are funded in part by Genomics Australia. So if anyone wants any more details, please head to our website or you can contact us through these channels here. So in terms of what we offer, we're open to all research disciplines. We have a collaborative screening model, so we do a lot of fee-for-service given our geolocation in Canberra, as well as, of course, because of COVID. We consult with clients about screening design and assay workflows, and we also assist with grant applications. So if you want to put a screen in a grant, you can come and chat to us. We also enable access to biological or chemical libraries. For screening, these can be compound libraries and also functional screening libraries, which I'll touch on a little bit more in the next few slides. Of course, we also provide general screening consumables and reagents for purchase. People think of us as a high-throughput screening lab, but of course, we also need to perform assay development. So we help screeners help develop their assays and do pilot experiments, which are proof of concepts for their work. So we do also require a lot of different instruments to do this. So we have instruments to measure assay readouts. So this includes high-throughput plate reader, a high-throughput or high-content microscope, and of course a high-throughput FACS analyzer as well. We have liquid handling and dispensing robotics, which range from the precision liquid handling through to bulk liquid dispensing robotics and non-contact dispensing robots as well. We have microplate incubators and storage towers, which hold all of our microplates when we're screening, microplate bioprinter, and like the conductor of a symphony, we have automation, which brings all of this equipment together. And so just talking about the types of libraries available, I wanted to focus primarily today on functional screening libraries. So these libraries, I have these libraries in-house, and Kaylene has these libraries in the VCFG as well, and I'm sure she'll talk more about that. And also the Functional Genomics South Australia have access to these libraries as well. In-house, we have a short interfering or siRNA library and a single guide RNA or sgRNA library, both from Horizon Discovery. So these particular libraries are in array microplate format, and what we can do is, using our precision liquid handling robotics, transfer volumes of these reagents into cells, either through electroporation or transfection. Of course, we need Cas9 if we're doing CRISPR screening. And then over time, and with different perturbagens, depending on the workflow that we're doing for the client, then we'd have some sort of phenotypic acquisition. So these particular resources are available to all our clients and is co-funded by Genomics Australia. So I just wanted to give you a brief overview of what a cell-based screen might look like through our facilities. So we have microplates that we would use. We would generally then add our cells into these plates using our bulk liquid dispensing robots, so that we have cells that are represented in each of these individual wells in a microplate. Then we would select the reagent that we wanted to screen. So as you saw, you have RNA-based reagents, we have DNA as well, CRISPR, different compounds or drugs, and even viruses. And so then we would then transfer these in generally into our plates using this precision liquid handling robotics. Then after that, we would have some sort of assay time point or endpoint. So we might add different reagents, or we might stop the assay by staining and fixing the cells for downstream applications. There's all sorts of different things, and I'll talk a little bit more about this in the next slide. And then we have our readout we need to capture. So that might be a high throughput plate reader. So this is sort of the traditional way of doing high throughput screening, through to this high content imaging and fax analyzer based readouts, which really facilitate the phenotypic screening. So I guess in terms of phenotypic screening, we fully, these are all automated. So we develop all of these algorithms and quantitative approaches to measure your biology, and we do that for every individual client. And so the types of measurements that might be include very basic cell number or viability readouts. In a 3D context, we might be doing colony counting or measuring the size of the colonies, right through to more complex readouts, including morphology analysis, looking at motility of cells, looking at the subcellular localization of specific molecules, intensity measurements, which is quite popular, looking at translocation of different transcription factors, for example, intracellular spot counting, and even for the neuroscientists in the audience, neurite length and cell projections. And so here are some examples of some 2D images that we've been able to take. So there we've got cell morphology, live cell RNA staining, and also DNA damage staining, where the individual green dots here represent areas of gamma H2AX foci. So you can actually quantitate these specific dots and do readouts that way. And of course, these 3D imaging examples where we're able to grow tumoroids, in this case, these are lung cancer and brain cancer. And we've also been able to image Drosophila with GFP reporter eye, so the eye disc is green, so we can actually quantitate that using our readouts. So having an interest myself in the nucleolus and nucleolar biology and understanding ribosome biogenesis, I just wanted to show you a little bit of a vignette into what we do and how it can be applied. So in this case, we're interested in looking at specific drugs that disrupt the nucleolus. And so we're able to take some cells, we're able to treat with drug. We can then fix and stain using nuclear and nucleolar markers, and then image on our high throughput microscope. And then here is just some representations of different phenotypes we see. So this includes when leptomycin B treatment, we see these nucleolar ring structures. And when we treat with actinomycin D, we see disruption of the nucleolus. And we can actually use our image analysis pipeline to discern the individual different phenotypes and then output these. And so this has been quite important for a number of cancer-related screens that we've been doing to try and find new therapeutics for obviously the treatment of cancer. So just before I finish, I just wanted to talk briefly about fax screening. So we do have flow cytometry-based readouts. We've just completed a screen doing combination therapies in heme malignancies. And so, of course, we can conduct cell-based assays using this instrument on the right-hand side here. We can also do immunophenotyping, bead-based assays, and also a combination of the two. So, you can really kind of flex these assays really quite substantially. And so you end up with readouts that look like this. This is a 384-well plate with different FACS profiles, which we can then translate into heat maps. And of course, if you're looking at the different combination screens, here we're looking at IC50 values of specific drugs. And then finally, I know Kaylene's going to talk a little bit more about this, but we're doing a lot more screening in 3D now, really because we want to better recapitulate the 3D microenvironment. This is particularly important for some types of cancer. In this case, we're able to grow spheroids, tumoroids, can do co-culture assays. We're moving into organoids now, which Kaylene will talk more about, and then matrigel and other hydrogel matrices. And these are just a representation of the types of things and readouts that we can capture using our instrumentation in our lab. So finally, I just wanted to thank my team and the ACTD, our vendors who make everything happen and keep all of our equipment in tip-top condition, our funding bodies that help keep our lab running. Here are our details if you want any more information. And I just wanted to briefly say that we're hiring 2 new senior technicians in our team. That's finishing on the 18th of September. So if you want any more information, please contact me or head to the Jobs AME website. Thank you.
Thanks so much, Amee, for the amazing talk. Over to Kaylene, our second speaker, if you want to share your screen.
All right, thanks, Cam. I should have titled this one same, but just a little bit different. That might have been the better way to follow on from Amee, who's got a great introduction, and really we have quite similar platforms. So I head the Victorian Centre for Functional Genomics, which is about to clock over 15 years in the making, and I have a joint appointment through the University of Melbourne in the Biochemistry and Pharmacology Department, as well as my substantive appointment at Peter Mac. Oops, slides are not going on. So what do? We are very much the same as Amee. We title ourselves as a genome-wide or targeted phenotyping and drug screening facility. We've got a team of 9 who you'll see on the next slide, and we're really highly experienced in driving innovation, technology and method development, and we have a large platform of infrastructure and reagents, and our whole working philosophy is to provide you with a complete end-to-end service, starting with your grant, starting then with assay development, guiding you through all of those steps, and most importantly, providing you with the bioinformatics data analytics readout at the end. So you don't have to walk away with a huge amount of data that you have to think about what you do. So like Amee, we've got a lot of screening libraries and resources. We do work in both 2D and 3D settings in all of these modalities. We've got a big suite of imaging infrastructure and a lot of liquid handling automation. And our platform is set up in a very much a modular format. We've got lots of different rooms, lots of standalone gear. So we have a lot of capacity for people to work with us at any time and at any level. And then we have a customized image analysis platform. That means that every project we do is relatively unique, and we work with you for the best readout. And then we manage everything, every single project through a Confluence portal that we call Prime. Here's our team, little snapshot of everybody on the side here. So as I said, we're 9, we're 4 postdocs and 4 people who just didn't do PhDs. They're amazing, and many of them have been with me for a very long time. And I can't survive without Louise, who is my admin assistant and just takes care of everything I need to know every day. But you can see here, just by looking at the team, that everybody's got a domain, but right behind that domain is absolute redundancy at the 2nd, 3rd, and 4th levels. So come into our lab, we train you, you work with us, we do fee for service as required, but typically that's interstate or someone who might drop cells off on a Monday and come in on a Tuesday, and we might finish that for them on a Thursday. But we work hand in glove with everybody. So we know all the extent of your project. We can often help you with add-ons, value adds that you mightn't have thought about, that simply comes from the experience that we've already got. but you would talk to me, and then you would talk to any of the team members that are relevant to the project. And sometimes 4 different people might actually be part of your project. You know, Carla is our data analyst. She actually doesn't do too much in the way of experiments anymore, but she'll help you get set up on the microscope, for example. Then she might analyze your data. Susie might review some of that data, even though she does 3D data analysis, because we might be trying to innovate in a new data analytical method. So the message here is we're a big team, but we're all here to support you, and a lot of different people will touch hands with you on the way through any project you do in the lab. Amee gave you a nice workflow of how we do our screens. This is just a snapshot of the way we look at it. Liquid handling infrastructure, delivering our libraries. We get our libraries from Compounds Australia up in Griffith University, so they are an incredible support for us. Assay ready plates come to our experimental design, a terrific partnership we've had with them for many, many years. And we deliver our CRISPRs and our RNAIs through liquid handling robotics. We then acquire our data, and that can be high complexity, low complexity data, whole well data, individual cell data, you name it, you can do it. And that could be through either image-based readouts, plate reader-based readouts, or fax-based readouts. And Amee mentioned the IntelliSite that she has. We don't have one of those here, but we have one at WeHi that we can access, or else we're also branching into using imaging cytometry as part of our suite of readout options. So there's a lot of choice right through from confocal to bright field to live imaging, all in high throughput setting, and then down to analysis, where when we talk to a screener, they'll say, oh, I want to look at my hit list. And we'll say, no, actually you don't. We want to look at how good your screen actually is. So we spend a lot of our time at the front end working on fundamental QC, percentage of variance, statistical reproducibility, dynamic range of your positive and negative controls. All of that must happen before we can say anything about what your screen readout is. So that's really, really important. And then Amee alluded to the large scale types of analyses that we do. And I just thought I'd give you an extension flow of that and how that looks. And the modalities that we use are very much similar. We don't have a Perkinoma microscope. We have Celomics instruments, but we use the Cell Profiler third-party software from the Broad Institute, which is highly customizable. And the whole strategy that we use is a cell phenotyping pipeline, or cell painting, where we identify the nucleus and the whole cell body here. We can do that in 2D and 3D settings. We then use the Cell Profiler software to identify cell features in all different metrics. And many of them have names that mean nothing to us, but they're computationally derived, meaningful information about the cell dynamics, whether it be the intensity of your stains, changes in shape, texture and topography, how close they are to each other. The same way you can look at that in a 3D setting. Are they round, elliptical? Are they tightly packed, loosely packed? All of that goes into the wash and comes out in some type of phenotypic signature. And you can see here that we're only looking at things that correlate with each other and are statistically different from the controls. And we generate these little signatures here, and you can see that they basically define different types of cellular connections. From there, we go back to the good old days of biological information. We're really integrating other screens, so we don't sit just looking at our own screen in totality. Other cell types, other screen readouts, GWAS data, clinical data, all of that needs to be embedded with your screen to give you the capacity to triage from a lot of targets down to a subset of very few that you would follow in greater detail back in your own lab. And just an illustration here using CRISPR screening, you can see our control here with the statistical range on the side here barely varies, looking at imaging features. And we've got 5 other different targets here that are the lead agent for classification of targets that all look like it. So just looking at BRD4, when we knock it out, we get this particular profile. You've got a very significant string of features down here that define that signature. Knocking out EGFR looks completely different. And you can see that the profile for it, oh, sorry, that's EGFR. The profile looks completely different. So we use these to group things to tell us which targets we should be looking at collectively. And very quickly touching on our 3D pipeline here, we embed ourselves in high throughput using either the rastrum or our Janus robots. So we're either using a hydrogel model or a matrigel model. We do that in a 384-well plate, and we've implemented a daily bright field imaging strategy, which is incredibly important because the depth of information we can get looking at structure formation over time gives us so much more than an endpoint readout of simply viability. And so we can do this in primary cells, patient-derived xenografts, cell lines, like Amee said, you name it, we can embed it if it's going to grow. A lot of that comes down to what you know about your cell model and how best it is positioned. But we can add our drugs here using a digital dispenser or the big robot over this side. And we're imaging primarily on our citation microscopes, either the citation 5 or the C10. And we use them for bright field and then for our final stages of fixed imaging. And then we implement that back into that pipeline that we just talked about, where we can then identify individual structures alone, do our custom analysis. This is really important because we do a lot of drug synergy interactions where we can have a profile that you can see that that drug combo at that dose point there gives us the most effective killing range. That's necessary, particularly when we're in a clinical situation where we're looking at standard of care treatment plus augmentation with something else. How's a patient going to respond? So we do a lot of that type of work as well. And I just thought I'd show you what this time-lapse imaging in daily bright field looks like, because it's absolutely outstanding to be able to see things over time. We can quantify each of these frames over time as well, but effectively we're looking at about 80% of the well in one field of view. And you can see our controls here growing happily over time. And you can see treatment with compound X at 10, 1, and 0.1 shows you significant death, less death, not that much death. I show this to illustrate as well that there's almost always going to be heterogeneity in the way that the lines form their structures, whether they're patient-derived material or cell lines, and we can actually distinguish and discriminate structure size and response to treatment by using imaging. So we can score all the big ones, all the small ones. We can then look at how did they respond to the treatment conditions. That in itself starts to get us towards how is a patient in a true heterogeneity setting going to respond to treatment. I'll put a little snapshot on the side here to just show you that we've been working very hard on methods for fixing and staining. It's no mean feat to do this in a high throughput manner, but we've nailed it. The structures stay in place, they stay the same size. It allows us to correlate all of our bright field time course with the stained endpoint. We can stain up to 5 different parameters. Typically, we're using cell machinery markers, and we can go and put them on our high content Cellular system and image individual structures. And you can see here how important this is. Paclitaxel and staurosporin both kill, but they kill completely differently. And you can see that once you've actually got cellular markers on there. We can then go and look for the agents that kill like paclitaxel or the agents that kill like staurosporin using machine learning strategies. So imaging at the end of the day, it wins every time. I just thought I'd throw a slide in to just talk about the things that we do, because as much as we're a core support platform, we love partnering with key champion labs that want to do something new. And so we're just about to submit our paper for publication on high throughput fixing and staining and multiplex imaging analysis. We are working with our sequencing platform right next door to me here on MaxSeq, multiplex analysis of cells. That's high throughput sequencing in a 384 format in 2D and 3D. That's a game changer for us. That's going to tell us how we can match phenotype and genotype. We do a lot of immuno-oncology studies. We do that in 3D settings with all ranges of immune type of cells. We also do it in the IncuSite, just looking at tracking death after putting in an immune cell setting. And we're doing a lot in machine learning to screen patient samples in that 3D setting. So our most recent example is 12 pancreatic patients, where Susie's then gone across all of those 12 and done the standard, well, it's not even standard, what you would think about as standard, but really quite difficult, artificial intelligence strategies, where we are looking at all the patients, finding commonalities, finding distinct differences. We've got resistant patients, sensitive patients, different looking structures. How can we relate those drug responses across different people? So we're using machine learning to do that. I mentioned already we do drug synergy interactions, and we also have methods developed for nucleofection of single guides out of our CRISPR library in 96-well format, only in 96-well format because the nucleofection system is in that setting. But this is really important for those people that are doing knockout in immune cells. So we can do it very easily in adherent cells. That's just a lipid-based transfection, but working in immune cells, really, really difficult, but we've got that methodology working as well. I'm not going to acknowledge my sponsors because, or not sponsors, but funding bodies and the fact that the vendors keep us alive because they're pretty much the same as Amee's. I just wanted to finish on a slide to alert everybody that Phenomics Australia, our major NCRISP funding partner for very many years now, has just put out a new consortia called in vitro disease modelling, of which both Amee and myself are part of, and it's accessible nationwide. It sits at a number of different institutes listed here with pretty pictures of all of us right there, really functioning on genome engineering and disease modelling through stem cells, through organ cell models like we just talked about in 3D settings, in 2D settings, in high throughput and in low throughput. So head to the Phenomics website. You can learn more about our latest series of funding and make use of platforms across the nation. Thanks, Cam.
Thanks so much, Kaylene, for the amazing presentation. I especially loved hearing about the citations in action and also hearing about machine learning and the advancements for patients. It's really, really cool. I haven't seen any questions pop in the chat, but I suppose my question's a little bit more open to both of you. My first question would be about pinpoint base editing. So we've got, it's basically more precise than CRISPR. And I thought I would ask what your thoughts are on this area, given that it recently launched. I'll hand it over to Amee if you've got any thoughts.
Oh goodness, putting me on the spot.
Sorry, Amee.
No, that's okay. I mean, look, I guess all these technologies, they are evolving. And so I guess where sort of Kaylene and I sit is really in the high throughput domain. So we are looking from a technical perspective, wondering whether or not we can do this sort of stuff in high throughput, right? So yeah. So that's sort of, I guess, I don't know, Kaylene, did you have anything to add to that? Or?
Well, I think that couldn't have been a better prompted question, Cam, because really, head to Phenomics Australia. We've got some incredible CRISPR platform resources there that can do this type of work. So again, with Amee, in part, we see a need, we try and fill a need in a technical capacity, but no one's coming to us to ask how to do that right now. And at a single target focus, we're probably not the best people to come to. It would be more like the Monash CRISPR organoid group or the Monash mouse modeling group, the Murdoch CRISPR and IPS group and Swalvatang's team, the WeHi team that are all doing these mouse models. Until base editing comes into effect in a more high throughput manner, it's probably going to be something that we would leave to those guys. That's their sphere of excellence. Cool. I would say, though, that I can see it circling back to us when cell lines get modified, and then there's a screening question to come in after that. We've sent cells out to Monash right now for a group to make a base edit in a target so that they can come back in and screen in drug libraries. So that is where I can see, it closes the loop.
Yeah. No, absolutely agree.
Right, amazing. Sharon, did you have a question?
Yeah, I was going to follow on with a discussion that Cameron and I were having earlier this morning about AI, and then Kaylene kind of launched into it, which is pretty interesting. And we're certainly seeing quite a few of the imaging vendors kind of launch into AI, some more successful than others. Kind of wondering what your thoughts are around that and how much you'll continue to use, or do you see it advancing more?
It's amazing. It's really hard work. Right now we've got a partnership because we generate so much data with a group in Europe who want to use their machine learning strategy across cell feature data to see if they can predict drug efficacy and specificity. I'm like, all right, great. We've got tons of screens here. If you ever got that, that's excellent. It's difficult, and because it requires you to train a data set, you train off the data set you generate. So you actually train on 80% of it and then go back on 20% of it. You can overtrain or undertrain. So getting that right is quite challenging. And this is not new. Machine learning, certainly out of the Broad Institute, has been going for some time. It's just a question of when's the right time to use it, perhaps, and how to use it. The work that Susie's done in the pancreatic is beautiful. And in fact, we found some clinically relevant compounds out of that we might not have found otherwise. And the challenge is now, unlike years ago when we would screen with one cell line or maybe two, most people are coming in with 10, 15. They want to look across a lot of patients now. Every person's different. So this, I think, is here to stay. It's a question about how we streamline it to make it more easily usable. And like anything, even at the level of sophistication of our cell phenotyping, still comes down to how do you pick the winner? Even if we can cluster things and look at the differences and group targets together, what's going to be the target group that we take away? Even from a machine learning perspective, you're not going to get one answer out. So it's still going to be how do you pick which one to follow up after that as well.
Great. I've got one follow-up question. I'm not seeing whether we've got too many other questions, but I've worked pretty closely with both of you for a really long time. And it's always fascinated me over, the course of the last 15 years when we've had that conversation of what do you see next? What do you think's coming? Where do you see, the next big thing? And, like 5 years ago, we were just starting to talk about 3D kind of analysis, and it seemed very difficult and almost unmanageable. And yet now both of you are, doing that, on a regular basis. What do you predict in the next couple of years? What do you think is going to be the next big thing? Amee, I might start with you.
Oh, goodness. Wow. Okay.
Tough one, right?
Well, no, I guess it's sort of, I think it will be an expansion of what we've discussed today, really, and better facilitation of that. So I guess in terms of the 3D modeling, and certainly being able to, use these sort of systems for patient diagnosis would be really, really cool. So I guess it's sort of moving towards that and just managing and being able to take all that phenotypic data that we collect and being able to kind of make it into something meaningful. I think with, I think that's just going to expand. So I think we're sort of in that phase at the moment where that's really sort of taking off. I don't know, Kaylene, did you have any extra thoughts on that? Or
I think the bottleneck at the moment is actually in the 3D setting is making the organoids from patients. We're all feeling like we're poised for a personalized medicine explosion, but the clinicians here still kind of go, I don't know, how do I trust that better than a 2D? I'm like, oh my goodness, this is not the way we should be thinking. So drug companies, everybody needs to get past that hurdle that a 3D setting is more robust and reliable. But beyond that, we need to be playing with organ on a chip. We need to be recapitulating the full microenvironment, the full cell complement, the whole shebang. That's where I see us going. That's taking us out of high throughput. That's very much funneling us down to sort of the bottom line where we've got targets. So I can't see us moving away from a tiered screening strategy where you may not screen all the compounds in an organoid setting. You might screen still in a 2D model, come to a 3D model, come to an organ on a chip model to more closely approximate that. I think something else I was going to say about that. Yeah, really, we've got to get these cell models working. That's probably the biggest challenge at the minute, I think, is to do that actually.
Great insights. Great insights. Thank you.
We actually do have a question from the audience members. Sally actually asked, can you elaborate on the scope of the RNAi assays with the siRNA libraries available?
Whatever you want it to be, Sally. We've done all sorts of stuff. We're putting RNAis into cells in 3D at the moment. That's a prelude to CRISPR, but the challenge of CRISPR is that you won't edit in every cell. So we've got to work out how to do that. I mean, we in all honesty, not many people use the RNAi libraries anymore. We use them for tech dev because we know exactly how they work and we know how easy it is to get them in. But by and large, there's a place for RNAi and a place for CRISPR. Most people want to use CRISPR. And because you have CRISPR-I, you actually have got a mechanism that it looks very much like an RNAi. But our libraries are still very useful. We still quite often, have people coming in and just getting a few targets here and there to test out something, but more than anything, people are screening in the CRISPR space. So when the question really that you came about was what sort of assays can we do? And really pretty much anything. It's a high throughput readout. We can multiplex stain. Amee showed the fax-based sorting. We've got image cytometry sitting there as well. We've got capacity to put these RNA into suspension cells and adherent cells and cells in 3D working in progress. So yeah, I think that's Amee and I live by the same motto. Just because we haven't done doesn't mean we can't. So
exactly. Dream big.
You've got an idea about what you want to look at. So yeah.
Amazing.
Actually, Sharon, one more thing I was going to say. The next big challenge, even though we talked about machine learning, is actually matching the genotype and phenotype, and then metabolic readouts and proteomics readouts.
Yeah.
So that's sort of like that is the one.
That's the holy grail, isn't it?
That is the one I forgot, because now we can do the sequencing. We can do it. So now we have to work out what it actually means and how we relate it. So
exactly. Yeah.
Perfect.
Amazing. Well, thank you. I don't see any other questions pop up. And I just want to say thank you very much for both participating and for your answers and your time. So thank you very much for joining.